This is episode 220 of the Stem Cell Podcast, Developmental and Synthetic Biology, with Dr. Michael Levin. Hey, everybody, we are Doctors Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have Dr. Michael Levin from Tufts University on the podcast to talk about his research using endogenous bioelectric dynamics to enable new capabilities in regenerative medicine and engineering. We've also got our usual roundup of recent highlights and stem cell news coming right up.
But first, take your human pluripotent stem cell cultures further with M-Teaser Plus from STEMCELL Technologies, the most widely published medium for feeder-free human ES and iPS cell maintenance, is now formulated for enhanced performance and versatility. M-Teaser Plus reduces medium acidosis for more stable cultures all weekend long. To learn more, visit www.stemcell.com/mteaserplus. We've got a few fun roundup stories for you today. A couple of clinical things that we're going to lead off with here. One, and I'll get started with this one Nature Biotech paper that just came out in Nature Biotechnology, which is tangentially associated with stem cell biology. It's going to tie into the second Roundup paper that I'm going to talk about a little bit later. Both my Roundup papers today are about the liver, which is certainly one of the most regenerative organs out there. And that's certainly in the realm of what we discuss here is regeneration, organ regeneration, something we're fond of talking about. The title of this paper is Transplantation of a Human Liver Following 3 Days of Ex situ Normothermic Preservation. And I want to frame this with sort of a broad discussion. We know that there's a shortage of human organs out there for transplant. A few months ago, there was the big news story about the cardiac, the pig heart that was transplanted into a human recipient who was not qualified to receive a human heart transplant. And that was it's a big deal because it's perhaps opening up a door for a new cache, a new collection of organs that would be available for transplant down the road. Ultimately, that's what we're trying to get at. It's more organs to reduce the overall organ shortage that's out there, and also certainly the black market for organs, which unfortunately does exist. And so these folks, this is really a collaborative effort between a number of clinicians, surgeons, bioengineers, mechanical engineers who have been able to develop this ex situ preservation device for livers, where they're able to maintain the appropriate function of a liver that was otherwise going to be discarded. It was not going to be utilized for transplantation purposes because it was not deemed good enough for transplantation. This liver would have otherwise been discarded, but they utilized their unique ex vivo ex situ approach to preserve it for up to a week before actually implanting it into a patient who needed this liver, obviously after liver failure, and that patient's doing extremely well. So we can dive into it a little bit, more deeply here. So we know that these current organ preservation methods have a pretty narrow window. They say usually less than 12 hours to actually assess, transport, and implant donor grafts for human transplantation. And this is sort of the case for not just liver, but for other organs as well, heart included. You have to really, do these procedures pretty quickly. The organs are not stable super long term, and this is a really hot area of study. And so here they transplanted this human liver that was discarded by, they're saying, all centers that were, that were analyzing this in Europe. All centers discarded it. They said it was not good enough for use. They preserved it for multiple days using their custom machine that was able to basically diving a little bit deeply into the machine itself that was able to do the preservation. And this is actually an earlier Nature Biotech paper published in 2020. The cool thing about this particular ex situ normothermic preservation machine was that it can monitor in real time glucose levels, acidification, output of certain, metabolites and waste. It's a real-time monitoring system exclusively optimized for liver transplantation. And this is really a cut above what's out there for preservation of livers long term. And so that was kind of the basis of the machine that they're using. You can check out the earlier Nature Biotech paper to take a deeper dive into that. So they use that machine to preserve this liver for up to a week. The other cool thing was they actually uh, corrected that liver that otherwise would have been discarded, according to the other centers out there. They corrected and fixed up the liver. They actually saw that there was a tumor growth in the liver, and they actually managed to clean that up and clean that out. And they, optimized the liver even further using their machine. And they transplanted into the patient that otherwise really had no other options. No other livers were available except for this one, which is like a last resort scenario, sort of like what was happening in the pig heart transplant situation a few months ago in Maryland. And the patient did great. After this liver was cleaned up for a week using this machine and maintained using this machine, it was transplanted to the patient, and the patient's doing great. The patient recovered rapidly and developed a normal quality of life without further signs of liver damage, such as rejection or injuries to the bile ducts, even after a 1-year follow-up, right? And the other thing that sort of ties it into a stem cell story was that the normal liver function, the regeneration capacity, which is tremendous in the liver, as we know, and the overall proliferation of the cells in the liver was maintained and improved over that course of 1 month, 1 year after the transplant. So the liver is doing its thing, it's functioning appropriately. And it really is, I think, a milestone for potentially not just liver transplantation, but perhaps other organs as well. If we can maintain organs ex vivo for up to a week or even longer, 10 days, um, this hugely opens up the amount of organs that we can use for transplant. And I think ultimately that is the bottom line here. We need more organs for transplantation purposes. They have to be high quality organs. They have to be able to survive long term in the patient into which they're transplanted. So I think it's a really exciting paper in my opinion. I'm excited to see the next set of applications for this cool ex vivo ex situ preservation technology.
Yes, agree. As someone who, we do, we get ovaries from brain dead organ donors, and of course, the vital organs, heart, lung, liver, are the first to go and have priority. So I'm always at the end of the line with the ovaries. But it's important, that warm, cold, and warm ischemic interval there between when you get them out of the patient, when you get them into liquid nitrogen or whatever freezing protocol is really critical. And we have to get under 4 hours. So it's always a mad dash. I can't trust a messenger to do it. I got to go to the hospital, stand by with a bag of ice. And when finally the ovaries come out, I race back to the lab. So someone who appreciates how critical the time intervals are here, I can say this is a major innovation. But just a couple comments. One is, how about being that patient, huh? I know, thanks God, the outcome in the end is all good, better than good. But like, imagine having that conversation where you're about to get the liver that fell off a truck and has a tumor in it. But as I said, the outcome was good, and this is, while I think ambitious, and I wouldn't say risky, I'm sure it was all within the realm of controlled risk. I wouldn't say it's lucky, it's fortunate that we had this positive outcome, because if it had gone the other way, I think it would have maybe cast a pall on this technology. And it really is incredible technology, as you said there, out to 10 days potentially. That's nuts. And as you also said, with the ability to kind of intervene and correct, fix up these organs, it's a big deal. And I have the same question as you at the end of it: is this system, this ex vivo perfusion system that they've pioneered here, it seems like it's very specific to the liver in terms of the monitoring and all that. But you wonder if a similar device, or the same device even, can be applied towards the preservation of other vital organs for that critical interval in between getting them out of the donor and into the recipient patient. So I'm really excited just to see how many patients will probably be saved, how many vital organs, livers in this case, will be utilized instead of discarded, and more excited to see how this technology is extended. So excited all around, Arun.
Very, very excited and agree with you entirely. I mean, I think the dream is to utilize this really cool device for other applications, other organs, heart, kidney, so many other things you can think of. And I think certainly every single organ has its own optimal growth conditions, optimal maintenance conditions. And, I'm not a surgeon, of course, and I'm not a bioengineer, but, certainly I think these things can be optimized. And I'm sure folks around the world are working on this kind of thing. And I guess to your first point about, from the patient's perspective there, considering this liver that supposedly quote unquote fell off a truck, right? I mean, if the alternative is death, I mean, you got to take it, right? And it's the same parallel that we saw with the guy who received the pig heart. The alternative was death. They were not going to survive if they did not receive that pig heart. In this situation, this particular patient is doing great after even a year. And that's speaking to the power of liver regeneration, the incredible regenerative capacity and the ability to self-restore the liver, right? Even this liver that had a tumor in it at some point. The cardiac patient was not as fortunate. Unfortunately, as we know, the heart doesn't regenerate as well after a heart is placed into the body, especially a pig heart. That's sort of a different situation entirely, right? That's a xenotransplantation approach. This is a human organ, so that's survival difference is perhaps attributed to that. Unfortunately, the pig heart patient, passed away after I think about a month and a half or so after receiving the experimental heart. But hey, lifespan extended, quality of life extended for a month and a half. Was able to interact with his family and friends, which is a great thing. So yeah, we'll see what happens with this technology. Perhaps there's other great applications for it down the road.
Yes, agree. And your comparison there is apt, but I think it also underscores, considering that's very experimental xenotransplantation, and this is here now, and I think it's underscored by the outcome there. This patient is back to life, and presumably has a really favorable prognosis. So very exciting times. And I'm moving from one amazing clinical innovation here that we just spoke of to another. I would say maybe the clinical intervention innovation that maybe defines this generation, at least with respect to cancer treatment, we're talking about CAR T, genetically engineered T cells that can target tumors. They've shown really amazing potential and anti-tumor activity in treating hematopoietic malignancies. That's pretty much a cure in many cases. Most cases. So that's been amazing, but the technology has limited efficacy in treating solid tumors. And that's been the horizon that I think everyone's been looking toward and trying to make progress toward. But the limitation there is, and generally with CAR T, is that they have a really poor capacity for in vivo expansion. And so they need to condition the patients. They need to give them this chemotherapy that can be toxic. It can kind of rule out a lot of patients altogether. And even when you do that, chemo kind of conditioning, the T cells that do adopt, that do engraft and become integrated, they terminally differentiate and ultimately become exhausted and dysfunctional. One approach that's meant to extend the life of these cells in vivo is to reprogram them with a kind of stem cell phenotype so that they have more renewal potential, extend their lifespan. And the existing approaches to that end, though, are confined to the manufacturing phase, right? When you have the cells in hand and you can manipulate them, once they go in the body, they're kind of out there. You can't influence them without influencing the rest of the body. And that's where this approach of orthogonal cytokine receptors came in. And that's a way that you can dose a patient with a molecule that'll have a specific effect on a target, and it won't have any off-target effect. The way you do that is, in this case, you take these cytokine receptors, the classic one that's been demonstrated is IL-2 receptor, and you mutate it so that there's a mutant form of the receptor that only binds a mutant form of the native cytokine. And the mutant cytokine doesn't bind at all the endogenous form in the body, and neither does the endogenous form bind the mutant receptor. So you get this one-to-one relationship with these mutant cytokine receptor pairs. In this case, this was kind of a technical story, I would say, with, I think, a lot of potential, tremendous potential. It's a bit of a nuts and bolts story, not necessarily pioneering any concepts, but it was a powerhouse group that was led by Anthony Ribas, Carl June, and Christopher Garcia, in between UCLA, Stanford, Perelman School of Medicine, where all those guys are housed. And of course, the common denominator here being the Parker Institute for Cancer Immunotherapy. What they did here essentially is they created chimeric receptors that hooked up that orthogonal apparatus with the mutant IL-2 cytokine, except instead of firing up the intracellular domain of the IL-2 receptor, as it would normally do, even in an orthogonal relationship, they created these chimeric receptors to test the ability of other interleukin receptors, in this case, IL-4, IL-7, IL-9, and IL-21. So they took the intracellular domains from those IL receptors and they fused them, making these chimeras so that they would be activated by this mutant IL-2. And what they showed effectively is that the one candidate, the IL-9 receptor intracellular domain chimera, was robust. It caused activation of STAT1, 3, 5, and caused the T cells to assume characteristics of stem cell memory and effector T cells. And as you might expect, this treatment had superior anti-tumor efficacy in 2 mouse models that were solid tumor models, melanoma and pancreatic cancer, even without doing that preconditioning, that chemo conditioning. So I think this is, I mean, while it is a bit of a nuts and bolts story applying previously identified concepts and approaches, I think this is representative of how we're tuning and hacking these systems to generate very robust therapeutic tools that, in this case, may get over the hump of applying the CAR T technology toward the treatment of solid tumors, which I think is perhaps not the final frontier, because the idea of using CAR T for everything, as we've spoken about, from fibrosis to targeting anything in the body. But I think in the treatment of cancer, solid tumors for CAR T is the final frontier, and I think we're one step closer with this big study from Nature.
Absolutely. Great paper here in Nature. I mean, this is a real powerhouse team. Chris Garcia, who's an expert in all things synthetic biology at Stanford, Carl June, of course, one of the godfathers of immunotherapy. Very collaborative. Multiple major academic institutions across the US are partaking here, folks at UCLA, Stanford, Penn. And you're right. I mean, I think solid tumors are really the final frontier for not just CAR T, but really cancer therapy in general. How do you crack that shell of the solid tumor and get to the real juicy stuff inside and kill all those cancer cells inside? One thing you notice here is they didn't just use a system to target any old solid tumors. They targeted pancreatic cancer. All right. And we know pancreatic cancer is one of the most lethal types of cancers out there. I think what the statistics are, like, greater than 80% to 90% mortality at some point with pancreatic cancer. It's really a death sentence in a lot of situations, unfortunately. So it got me to think about, in this situation where you have efficacy in a mouse model and an in vivo system and targeting something like pancreatic cancer, you got to think this is going to be accelerated, at least accelerated to therapy, accelerated to clinical trials, by the FDA and other folks who are doing these clinical things, right? I mean, because as we've discussed in the past with other folks who have been on the show with, focusing on pancreatic cancer, options are still pretty limited. And to me, this seems really promising, and perhaps they're going to fast track this towards therapy and trials, don't you think?
Oh, for sure. And I think we're kind of in the final leg of the race here, I would hope, at least. And that's why it's such a high profile paper. That's why it takes such a conglomerate of these, amazing researchers and physicians to get this going. And I think this is really tweaking this tool just in the run up to clinical trials, which I would guess are right around the corner.
Yeah, for sure. I mean, anything related to pancreatic cancer and showing efficacy against it, like what they're showing here, is gonna get the green light. Absolutely. We're gonna go back to the liver for a second. That's my second, story. Both my stories have to do with the liver. One is more of a clinical story that I just talked about, right, with this ex vivo perfusion system for maintaining livers for super long period of time. But now we're gonna bring it back to more of the basic science, more of the stem cell side of things, and we're gonna talk a little bit about something we've been talking a lot about recently on the show, which is these single cell atlases. Okay. It seems like you would think that every single type of single cell atlas has been done. It's been done in the last couple of years, in part because of the amazing accessibility of the technology becoming somewhat cheaper. Multiple people around the world are starting to use it. We actually talked about on the last show the human cell atlas, right? Which the first set of papers for that finally came out in Science. These super high profile papers, cataloging all the different cell types found in the human body using single cell. This is a specific application of single cell technology towards liver regeneration. The title of this paper, which is in Cell Stem Cell, is the spatiotemporal program of zonal liver regeneration following acute injury. These are some folks from the Weizmann Institute over in Israel. First author is Shani Ben Moshe, and last author here is Shalev Iskowitz. So we know that the liver has this remarkable ability to regenerate. We just talked about it, right? This patient received this damaged falling off a truck liver, and that liver was still able to integrate into the body and restore its function, regenerate appropriately, and it's fully functional after a year, right? And even after acute damage, like in this particular situation, they were talking about acetaminophen, acute acetaminophen or APAP intoxication. So this is actually, and I did a little bit of research on the side here. APAP intoxication, acute acetaminophen or Tylenol intoxication, is the number one cause of acute liver injury, as you might expect. People unfortunately overdose on Tylenol or acetaminophen or whatever, and it can cause a lot of damage. And so what they're trying to show here is what's actually going on in the liver cells in response to that damage, and how does liver actually regenerate during that injury. How do the existing surviving cells upregulate certain transcriptional pathways to compensate for the loss of those cells that died because of the acetaminophen intoxication? And what they showed through a bunch of single cell analysis was that hepatocytes can proliferate throughout the liver lobule, right, during the post-injury process. But this creates sort of a mitotic pressure, is what they call it. And this is required to repopulate that necrotic pericentral zone in the liver. That was, where those hepatocytes, the pericentral hepatocytes died in response to the acetaminophen. Okay. There's like this select group of hepatocytes that's typically the ones that die first after acetaminophen or APAP intoxication, right? But then the other cells are upregulating certain gene pathways, fetal specific genes, actually, some that are pretty well known, even for me as a cardiac biologist, like AFP, alpha fetoprotein, CDH17. And those adjacent cells are reprogramming to a pericentral state. Okay, so basically the surviving cells are upregulating these ancestral, these primordial, immature gene pathways to reprogram to a pericentral state. And we've actually talked about this a lot, I think, on the podcast recently, this idea of de-differentiation in the body, natural de-differentiation, and how this can happen after situations of injury, for example, in this situation, right? And so the zonated endothelial and hepatic stellate cells, or HSCs, and macrophage populations are differentially involved in the immune recruitment, proliferation, and remodeling of the matrix. And then there's this massive infiltration of myeloid cells in response, in accordance with a big decline in global antigen presentation. So I think it really comes down to that compensatory mechanism that's happening in the liver. So the targeted cells, the cells that are targeted by the acetaminophen intoxication, those are the ones that die, but really the adjacent cells are going to take over and do their job to compensate for lost cells by quickly activating, and that's another point, quickly activating these fetal transcriptional pathways that are found in the liver. compensate for the loss of the cells, take over, proliferate, and then this is really the basis for this astounding regenerative capacity in the liver. Not only situations for acute, intoxication, acute liver injury here, or perhaps more chronic conditions as well. I think the chronic it's a tougher nut to crack, right? This idea of chronic liver failure through constant alcohol exposure that can really limit some of the amazing regenerative capacity of the liver. But at least in acute situations like APAP intoxication, acetaminophen intoxication, these residual liver cells, hepatocytes, can really do a great job in restoring the overall function of the healthy liver.
Yeah, the maladaptive chronic injury type stuff, that's tough. And how do you get at it with single cell, given the long interval over which that takes place? Here, the real benefit of this model is that it takes place over, I guess, 4 days or something, that they're able to look at really discrete time points. And this, not enough, I think, attention is paid to this mode of liver damage and the therapeutic inroad there, because as you said, it's the most common. And also in terms of cell therapy, it maybe is the earliest best application of like liver organoids, for example, because you could have like a modular off-site mini organ that could perform the essential functions of the liver. And in that case, you wouldn't have to have a long term, right? You just have to have them bridge the gap while the liver is recovering and regenerating, so that you don't die. So I like the model that they used here. My only question here about the study as a whole is that as soon as I saw zonation in the title, I expect to see a lot of spatial transcriptomics, especially that they're going single cell. And I didn't find any. I'm assuming it's not in the supplement. They would have brought it up there in terms of the profile, but I'm wondering, I'm guessing it just hasn't got there yet, but I'm expecting to see a lot of stories using spatial transcriptomics in the liver, because the architecture is so critical to the regenerative function. So I'm looking forward to seeing that from this group or others.
No, I think there actually was a bit of spatial transcriptomics that was conducted here as well. Perhaps really it wasn't the major focus, but I do think there was some done here. But I think I agree with you. I think this is a very specific approach, very specific targeted injury and acetaminophen intoxication. And the other general question I have is, how applicable would this particular data set be to other situations of acute liver injury? I think there's other examples of that too, not just acetaminophen. And certainly acetaminophen as a drug, a small molecule, can really detrimentally impact transcriptional activity in these cells in a very targeted, specific way. I don't know if it's just the death, right? But perhaps the drug itself is also having a transcriptional effect. So as we know, small molecules do, small molecules have a bunch of off-target effects. So I'm curious to see, what about another targeted acute injury of the liver? Are we going to see some similar transcriptional impacts? And the other small caveat here, just mentioned the limitation. These samples are apparently taken every 24 hours, which doesn't necessarily get the transcription, the temporal resolution that you would want in such a rapid situation of regeneration, like in liver. Perhaps, something like every hour or every few hours, you can get a sample and do your transcriptional analysis, right? But that's if you have an unlimited bank, which we don't, right?
Well, Arun, you got that startup money. Go get after it with 10x. But you're a heart guy, so I guess you'll leave it for them. One segue I have here is that, this is you mentioned the reactivation of a fetal program, and that's going to play into my next story here, although not for a regenerative purpose, but more of that maladaptive disease function. I'm going to come back to that. But first, just a brief intro. This is about pulmonary hypertension, okay, which is frequently associated with heart failure. Arun, you must know. And it can occur from many different causes. So you can have pulmonary vascular disease or heart or lung disease. You can have pulmonary embolism and other causes as well. But it's characterized typically by progressive, sustained increase in the pulmonary vascular resistance and arterial pressure. And that leads to reduced cardiac output, culminating ultimately in right ventricular failure and death. When pulmonary hypertension develops specifically in the pulmonary arterioles, it's called pulmonary arterial hypertension. And that's characterized by vascular remodeling, including neointima development and these complex plexiform lesions that form and ultimately occlude the precapillary arteries and lead to their loss. So vascular remodeling is a hallmark of pulmonary arterial hypertension, and that results from obviously an alteration in the vascular cells, right? Their transcriptional program and their behavior. And indeed, if you take vascular cells from a patient with pulmonary arterial hypertension and culture them, the phenotype manifests and is maintained for a sustained period outside. So it looks like it's a cell intrinsic issue, at least once the disease is set. But the regulatory mechanisms that cause the normal vasculature to assume this bizarre phenotype are elusive. They remain elusive. It's known that the cells change, right? But the mechanisms of how they change is not really well understood. There is a hypothesis, of course, and that's voiced by Soni Savai Pulamsetti, sorry for the mispronunciation of the name, who is at the Max Planck Institute for Heart and Lung Research. And her group hypothesized that the persistent phenotype of these pulmonary arterial hypertensive vascular cells results from alterations in histone modification and associated epigenetic changes. So they went after this hypothesis using, I think at this point, a very straightforward approach. RNA-seq and ChIP-seq. They took the pulmonary arterial fibroblasts from a patient with pulmonary arterial hypertension versus control lungs. They show that there was, thousands of differentially transcribed genes. That's not really a big surprise. They also showed there's a lot of different chromatin accessibility, for over 4,000 active enhancers that are altered in the patient samples. But ultimately, they settle on integrating these two approaches, and they identify a signature that shows that the transcription of genes that underlie lung morphogenesis were de-repressed in the patient samples. So morphogenesis, things that typically take place during fetal stages, and that's how I led there. Coming back to that, it looks like there's this activation of TBX4, TBX5, SOX9, again, all usually expressed during morphogenesis. And that seems to contribute to the phenotype, because when you silence these factors or this E1A associated protein, P300, using RNAi or small molecule inhibition, you get regression of the PAH, pulmonary arterial hypertension cellular phenotype, regression of that mesenchymal signature that the cells display. And here, I think this is why it was a science translational medicine story here. And, I think a stem cell story in that it's going to the root of disease here. But it was a clinical and translational story here, because they actually showed that if you use pharmacological inhibition of this P300 protein complex, you can reduce the phenotype in 3 different models, rodent models of pulmonary arterial hypertension. And finally, they take these precision cut slices from human pulmonary arterial hypertension lungs and culture them ex vivo in the context of this compound, and show that it can also reduce the phenotype there. So this is, at this level, when you're reading papers in these big journals, there's all kinds of tech crazy that goes in there. Each paper costs hundreds of thousands of dollars. I think by contrast, this was a really more straightforward, I would say, economical study that used methods that aren't, cutting edge, but are still advanced enough to give resolution of this phenotype in these cells, and specifically, most importantly, in these patient samples, with a treatment that may actually be eligible for clinical trial. We'll have to see about off target, but could go into trial relatively soon. And at the very least, I think understanding the mechanism by which the phenotype exerts itself, this reactivation of a fetal transcriptional program, I think is an important conceptual advance. So Arun, I don't know what you think, but I really enjoyed the story.
Yeah, I enjoyed it too. I think there's a few different reasons as to why I enjoyed it. One, as a cardiac biologist and a cardiac developmental biologist, it was cool to see TBX5 mentioned because that's all obviously a very important gene for cardiac development too, but also looks like it's important for early lung development, which I don't know as much about. But yes, I think the power here is in the models. This is like you said, a pretty economical study, just RNA sequencing and ChIP-seq of primary samples. And then from there, really validating the pharmacological intervention and the treatment, quote unquote treatment in these multiple mouse models. Okay, rat models, three different rat models, or maybe rodent models. I actually don't know if they're a mouse or rat. Three different rodent models. And also this ex vivo slice culture from pulmonary hypertension to validate your observation in multiple different model systems and relevant model systems too, in vivo and also human model systems. I think that's probably why this is a science translational medicine paper. One little thing to mention here, I'm actually very happy that they didn't consider iPSCs at all in this particular paper. Maybe that's very taboo of me to say since I'm an IPS modeling guy and we talk about iPSC modeling all the time on this show. But this is one of those situations where you don't necessarily need iPSCs to model pulmonary hypertension. And I think in this situation, it probably wouldn't tell you much, right? Because as we know for iPSC derivation, it screws up and, changes the entire epigenetic landscape, which is what they're trying to maintain here with these primary cultures of the fibroblasts from PAH patients. So iPSCs aren't good for everything, Dale. That's what I'm saying here. They're good for a lot of different things, but in this situation, I'm glad they didn't use them.
Yeah, I mean, I guess lacking any kind of embryologic or developmental angle, although it is a reactivation of this fetal program, I think that it makes more sense here to go right to the patient tissue if you can get it. And that's what they had the opportunity to do here. And I think that also is why this was such a high profile paper, is because you're treating tissue where the phenotype has already exerted itself. And there's no real discussion of whether or not it's reversing the disease, right? A cellular phenotype ex vivo, even with the substantiation of the rodent models there, I don't think is enough to say that we can be putting this into patients right away. But at least as an idea, I think it's really important to show that patients who are already affected by this condition maybe could find some help. So an exciting story, as you said, economical, but a major impact. So moving on to the interview with Michael Levin in just a minute. But first, I have a message from STEMCELL Technologies. Neuroscientists looking for more predictive power in their disease models are increasingly adopting human pluripotent stem cells in their research. STEMCELL Technologies offers products, protocols, and training to support HPSC-derived neural models. Explore their collection of technical videos and webinars on neurological disease modeling by visiting www.stemcell.com/neural disease model. All right, everybody, for this episode, we have with us from Tufts University, Dr. Mike Levin, who is director of the Allen Discovery Center at Tufts, also distinguished professor in the Department of Biology and the Vannevar Bush Chair. Dr. Levin is also associate faculty at the Wyss Institute. His group focus is on understanding the biophysical mechanisms that implement decision-making during complex pattern regulation and harnessing endogenous bioelectric dynamics toward rational control of growth and form. I don't know what that means either, but he's going to explain it to us. The lab's current main directions are understanding how somatic cells from bioelectrical networks for storing and recalling pattern memories that guide morphogenesis, creating next generation AI tools for helping scientists understand top-down control of pattern regulation, and using these insights to enable new capabilities in regenerative medicine and engineering. Dr. Levin, thank you so much for joining us today.
Thanks so much for having me. I'm happy to be here.
Yeah, thanks so much for being here. And let's actually start off by taking a bit of a step back and talking about some of your first forays into developmental biology a few decades ago. You actually did your PhD and postdoc at Harvard with Cliff Taven and Mark Mercola, respectively. Dr. Mercola was actually on my thesis committee, where you performed the first characterization of some of the mechanisms that allow embryos to consistently form left-right asymmetric body structures, really focusing on the activin, sonic hedgehog, and nodal signaling pathways. The work is actually on Nature's list of 100 milestones of developmental biology of the century. So that's pretty cool. Walk us through kind of that time in your career and what really inspired you to work on such a fundamentally important mechanism in developmental biology, this idea of left right asymmetry that you've kind of stuck with in your career for a really long time since.
Yeah, my work in developmental biology actually started even before that. As an undergrad, I worked with Susan Ernst, who was a professor that studied patterning in sea urchins. And some of the very first research in that area I did by looking at applied magnetic field effects on sea urchin development and trying to ask how external magnetic fields could interface with some of the biophysics that drives development. And then in Cliff's lab, I fundamentally was interested in development because we all take this journey from something that some people might call quote unquote just physics or just chemistry. So, an unfertilized oocyte, a collection of molecules, to a complex cognitive being with goals and preferences and hopes and dreams and so on. And so that slow process of how physics becomes mind is, perhaps one of the most amazing mysteries that any of us can work on. So that's what drew me to embryonic development, and I wanted to work with Cliff because of a talk actually he gave at the first year in grad school. And in his lab, it was kind of somewhat fortuitous that this amazing project became available, where through Cliff's and his postdocs work on sonic hedgehog, they worked on it in the limb and in the neural tube. But actually, there was also some data showing that it was asymmetric in the very early embryo, and this would have been the first gene known to be asymmetrically expressed. And as soon as I saw that, I said, "This is remarkable. We have to work on this because asymmetry, for many reasons, is really has fundamental interests. There's lots of physics and other aspects that are required to get left-right asymmetry correct." And so this kind of merged many of my interests, including physics, developmental biology, all of those things. So that's how I got into it. Yeah, turned out to be a fascinating project.
Yes, fascinating. And you touched on it there. What's so fascinating, miraculous, and humbling about what we do is that journey, right? From just physics, the molecules, to biology and tissues and organized structures that perform function, and then like transcending that to the mind. I think that's one of the first things as a biologist that really struck me and overwhelmed me with the complexity. And it was very humbling, and I think we're in a similar place with therapeutics and cells, right? As cell-based therapies move closer to reality, there's still significant challenges to construction of tissues at practical scales, or maybe call them therapeutically applicable scales in more traditional paradigm of organ tissue transplantation, let's say. And of course, everything's a bit of a hack when you're working with in vitro cultured pluripotent stem cells, that don't really have a correlate in biology. But I have been impressed with approaches like that of Matthias Lutoff, who combines this kind of cell intrinsic morphogenic momentum, let's call it, with this extrinsically enforced physical scaffolds. It's this combination of the cells doing what their program is and also kind of engineering it. Having tissues that are both made and grown, so to speak. So given that intro and that tremendous journey from physics to biology to transcending in the mind, and also a similar journey we're taking from cells to actual practical tissues, and given your experience with pattern formation at the earliest stages, as you alluded to with left-right asymmetry, how do you envision tissues and perhaps even someday organs being made from cells in like a really practical way? What's your vision of how that's going to happen?
Yeah, my vision it has a lot to do with really this very deep invariant between the question of mind and the question of pattern. And I'm certainly not the first person to think this way. You know, Alan Turing, who's one of the forefathers of computer science, was very interested in artificial intelligence and in mind and in various embodiments of mind. And he also had this amazing paper on self-organization of morphogenesis, right? Of so-called Turing patterns. And you know, you might wonder, why would somebody who was interested in computation and mind be interested in morphogenesis? And I actually think that's not a coincidence. These are fundamentally the same problem, I think. And the way I think about it is that morphogenesis is basically the behavior of a collective intelligence in anatomical space, whereas modern brains and nervous systems guide the movement of bodies through three-dimensional space. Those tricks came about by pivoting something that evolution learned to do long before that, which is to move the configuration of the body through morphospace. So anatomical morphospace being all the possible configurations of the anatomy. And embryos have to, they start out in the quote unquote wrong region of morphospace, and they have to get to the correct region corresponding to their proper species specific target morphology. And so this becomes a navigation task, and this is a navigation task performed by a kind of swarm intelligence of these cells working together. And what's really cool about embryogenesis, regeneration, remodeling, metamorphosis, all of these cancer suppression, all of these things have one fundamental thing in common, which is a kind of anatomical homeostasis. It's a kind of ability to start off at the wrong place. In fact, not even necessarily at the place you expect, which is to be a single cell. You might be a salamander limb and you might be amputated anywhere along the proximodistal plane, or you might be a newt whose early embryonic cells are made far too large, and they will still basically make a correct newt with the correct size by adjusting the number of cells, and in fact using all kinds of neat tricks to compensate for the fact that the cells are too big. There are numerous examples where basically what the biology is able to do is to deploy a kind of intelligence, and I don't mean that sort of in quotes or metaphorically. I literally mean intelligence in the sense of being able to solve problems. In fact, navigation problems. There are lots of roboticists working on swarm robotics, where groups of motile robots are able to solve various problems in space. And this is, evolution figured this out a really long time ago. And so I think that where really the promise of regenerative medicine is going to be fulfilled is when we move from the current approaches which treat the cells as hardware, right, and seek to really micromanage the process by giving them appropriate signals and getting, you know, 3D printing and all these kinds of things, moving towards a realization that we're dealing here with an agential material. This is not just a structural material the way engineers have been working with for hundreds of years. This is not even an active material or even a computational material. This is an agential material. These cells used to be independent, free-living animals. They have their own agendas. They have their own capacity for behavior. And the most important thing they have is the ability to merge together in groups to form larger scale structures that solve bigger problems. So you have single cells that solve single cell problems, things like metabolic homeostasis and so on. And collectively, they solve bigger problems like navigating in morphospace, starting off in various diseased or damaged or kind of early stage conditions and moving to where they're supposed to be. And so I think that much like computer science took the leap from being, you know, in the 40s and 50s, the way you would program a computer was you would physically rewire the hardware, right? That you can all visualize what that looks like. I have a slide that I always show in these talks where, you know, this woman sitting there, she's literally pulling wires in and out to get the computer to do whatever it's going to do. But now on your laptop, when you switch from Photoshop to Microsoft Word, you don't get out your soldering iron and start rewiring. And the reason you don't do that, right, is because computer scientists figured out that if your hardware is good enough, and I'm going to claim that biological hardware is definitely good enough, what you can do is you can control its behavior by stimuli, by inputs, by communication with it, as opposed to micromanagement. And you can take advantage of the intelligence, the problem solving of these cell collectives to do a kind of behavior shaping more than micromanaging or more than engineering directly. And I think that's going to enable us to get to large scale morphological outcomes that are going to be very difficult to reach through basic stem cell biology, CRISPR, genomic editing, those kinds of things.
Yeah, it's a perspective that we don't really touch on a whole lot here on the show. This idea of cell
Yep.
intrinsic intelligence. That's a very powerful concept. And, early development especially is really filled with examples of that cell intrinsic intelligence, going from a single zygote to, a complex multicellular organism. I actually wanted to get your perspective on a related topic. Actually, there's a new developmental technology that we talk a lot on the show that might end up interfacing pretty well with some of your work studying, actually, for example, bioelectric signaling and also some of these examples of cell intrinsic
Mhm.
intelligence. I'm talking about these human pluripotent stem cell derived blastoids, these analogs of early embryogenesis, the early blastocysts that are found during your early development, derived from induced pluripotent stem cells. They have a bunch of different applications. They've really caught on quite dramatically and quite quickly in our field. What do you think about the promise of that particular technology for studying human development, early human development, these artificial blastoids, and how well do you think they might be able to interface with the work that you're doing?
Yeah, I think these kinds of synthetic models are extremely interesting, and I kind of have an opposite view of this than a lot of people. Where you often hear these critiques at how well these things don't really recapitulate the in vivo situation, that are not really accurate, and this and that. Uh, I actually think that we learn the most from exposing cellular collectives to novel scenarios and novel environments that are as alien to their kind of in vivo expected situation as possible. That's where you really learn what the collective intelligence is capable of. That's where you really get to find out what decisions they're making, what measurements they're taking, what their capabilities are in novel environments. I mean, the basic point of intelligence is not simply that you can do the same thing over and over again, like we see in normal development. It's the center of this concept of intelligence is that you're able to handle novelty. You're able to handle new situations, new problems, to reuse the tools that you already have, whether they be the software tools or the hardware of the cell, to solve new problems and have adaptive behavior in new contexts. And so I think blastoids, organoids, the kind of synthetic biobots that we make, so like the Xenobots that we make, and all of these other things, are in fact going to teach us much more than simply trying to recapitulate the in vivo situation. I think that normal development really lulls us into a false sense of security, in the sense that it's pretty reliable, and acorns make oak trees, and Xenopus eggs make frogs, and so you sort of think, well, what else would it be? That's the only way it's going to be. But actually, once you start exposing these cells and tissues to novel environments, you find out that actually they can do a lot more. And you find out that, for example, the ectoderm of the frog embryo, those frog skin cells, their default behavior isn't in fact to be a boring sort of two-dimensional life on the outside of the animal, keeping out the pathogens. In fact, by default, when freed from the instructive interactions of the rest of the cells, they make Xenobots. They make these spherical self directed motile creatures that have all kinds of amazing behaviors, including replicating themselves in a way that no other animal does through kinematic self-replication, and so on. And so, yeah, I actually think these kind of synthetic models teach us about the most important thing, which is the ability of all of these things to handle novelty, because that raises not only it not only opens new ideas for roadmaps for regenerative medicine, meaning taking advantage of these capacities to coax the cells to build things that we don't know how to micromanage and won't know how to for quite some time. And it also sheds really interesting light on evolution, because what we're seeing is that evolution doesn't make specific solutions to specific environments and niches. It actually makes problem-solving machines that are able to solve all kinds of new problems they haven't seen before. So all of those things really are a great outcome of these kinds of synthetic biorobotics, organoids, all that stuff.
Wow. I mean, this is why I love doing this podcast. I'm listening to you, and I'm just thinking about whole different ways of framing the stuff we do, just with your language. And I
Yep.
think maybe your approach, the way you consider these cells as, having their own agenda and having intelligence. I love the term cellular collective. I'm not going to call anything tissues anymore. I'm going to call it all cellular collectives. Makes me think of the Borg. The idea that, I mean, is really exciting here, that's buried, or I mean, really on the surface of what you're saying here, is therapies, right? Is if you can get these cells to work on a more, I wouldn't say freewheeling, but in a more adaptable way that doesn't depend on our engineering, because we're no engineers like nature. So I love this idea of, it's kind of like regrowth, and I think it's really a great example of it is this exciting story you just had in Science Advances a few months back with this wearable bioreactor that you put on an amputated hind limb of a Xenopus, and it generated long-term functional recovery in Xenopus over the course of many months. This really has increased resonance for me, and probably Arun, since just a few weeks ago, we covered this story that was in Nature about terminally differentiated adult cells from human that were completely reprogrammed to pluripotency using only chemicals. So it really kicks open the door, this idea of, absent any kind of viral reprogramming or getting stuff into cells, working in the nucleus directly, at least, that you could just have a cocktail of chemicals. And that's very much what you did here in this bioreactor. You had a cocktail of chemicals that led to this functional regrowth. Is this kind of what you're envisioning when you talk about the therapeutic horizon, or is this like a really crude example of what we can expect to see moving forward?
Well, both. I mean, this is the amazing thing, which I don't know how clear that was from the paper, is it is a very crude example, because this cocktail that grew back after a 24-hour exposure, grew back legs in frogs. That was our first attempt at it. That wasn't cocktail number 72 out of a screen of 800 cocktails. That was the first one we tried. And so I've got to think that if we, as we perfect this, and if we really knew what we were doing, the optimal kind of future version of this is going to be amazing. Like this was just our first guess. And so I absolutely think this was a crude, just a very crude first step example of what's possible. I want to sort of talk about the strategy here, because what we didn't do was implant stem cells or continuously monitor and sort of adjust the leg, or 3D print any kind of pieces. This was literally just the intervention took place in the first 24 hours, and the effects to the leg grew for a year and a half. And the reason that we even tried this before is that I am firmly convinced that the roadmap to regenerative medicine is not through micromanagement. It's through finding the native subroutine calls of the software of morphogenesis. So finding out what are the triggers of specific kinds of very complex morphogenetic events that the body already knows how to do, and kickstarting those. Because again, as you know from computer science, yeah, you could write everything in machine code, but why would you want to when you have access to these high level libraries that the system already has built in? So I view our goal as trying to figure out what is the control structure of morphogenesis and how high up can we interface with it? Because what I would really like to do, and I think what we did here and what we did before in the tail regeneration work and so on, is not to say, here are the cell types you're going to need, and I'm going to directly differentiate all these cell types, nor here's the shape of the organ you need. All we literally said in that 24 hours to these cells is make whatever normally goes here. That's it. We, the cocktail that we used is not specific for legs. There's nothing leg specific about it. In fact, we previously induced leg regeneration using the same bioelectric drug that we used for inducing tail regeneration in tadpoles with spinal cord and muscle. And in both cases, you don't get a tumor, you don't get a kidney, you don't get a mess. What you get is the actual organ that belongs there. And so that's the trick, is not micromanagement. I want to, and just to give you a much more specific example, we study a lot. We study bioelectricity a lot, meaning electrical communication between cells. And the reason we study bioelectricity is because if you ask, how is it that collections of cells get together in a way that allows large-scale intelligences to emerge, right? We see that in the brain. So you and I are collections of neurons. These neurons somehow work together to have goals and preferences and memories that don't belong to any individual cell. They're a property of the collective. And we know, from neuroscience, we know, of course, that this is achieved through these electrical networks. Well, that strategy, evolution discovered that around the time of bacterial biofilms, using electricity as the cognitive glue that binds tiny competent subunits into bigger units that are competent about bigger things. And so we were interested in studying this hypothesis that these bioelectric networks outside of the brain could store patterns that guide the movement of this collective intelligence through morphospace, basically guide the movement of development and regeneration and so on. And so one of the things that we showed is that there's a very specific bioelectric pre-pattern that goes on in the early frog face that tells the face where the eyes should go. In fact, there's an electric face pattern that was discovered by my colleague Danny Adams when she was in my group, looking at that basically lays out all the features of the nascent face that then you can use to diagnose defects and in fact repair those defects and so on. And so there's a particular pattern that is what triggers eye development. And so we took that bioelectric pattern and we induced it elsewhere in the embryo. And the way you do that, we don't apply fields. There's no magnets. There's no electromagnetics. We modulate ion channels. So you can inject RNA encoding specific ion channels, or you can use drugs to open and close those channels and so on. So we inject a set of potassium channels in a particular location, let's say of cells that are going to give rise to the gut. And these cells basically get a bioelectric pattern that says, make an eye here. And they do, even though they're endoderm. Now, this was at the time, this was completely striking because we're told that only anterior neurectoderm is competent to be eye, and that's not true. You can make eye out of almost anything. And so bioelectrically, we induce that state, it makes an eye. And the amazing thing is, there's a couple of amazing things about that. One is when you inject, when you label the RNA so you can see exactly which cells got your channel, what you see is that some of the cells in the eye have your channel, and so you can see that those were the ones you affected. But they recruited a whole bunch of other cells that were themselves never actually targeted to participate in this eye formation. So there's two levels of instruction. We instruct the cells to make an eye. They instruct their neighbors as needed to complete eye formation because we didn't transfect enough. So that's the first thing. The second thing is that we did not provide enough information to make an eye. We didn't have to, the eye is a very complex organ, lots of tissue cell types. We didn't do any of that. We provide a very simple bioelectrical pattern that's basically a subroutine call that says make an eye here. So it makes an eye, the right structure, the right shape, the right size, and then it stops. That's the other kind of really key thing, that it's a self-limiting cascade. And so all of the downstream details as far as all the gene expression, PAX6, RIX1, all of these things, all of that gets managed downstream. We didn't have to worry about any of that. And that's my vision for regenerative medicine. Once we understand how to convince these cells of what they need to do, after that it's hands off and you let them do their thing.
So I think that's phenomenal. I think it's a great approach to perhaps integrate some of the amazing basic science work that you're doing with other approaches that other folks are doing in regenerative medicine. I think they can be complementary in a lot of ways. But I think, the Xenopus has really been a focus in your lab for a very long time. It's been foundational to the work that you're doing, driven a lot of the fundamental discoveries that you've had. And since you mentioned it, let's talk about Xenobots a little bit. Might as well. This is another technology that's emerged from your work in Xenopus. For the uninitiated, there are these in vitro biological quote unquote robots derived from the frog, Xenopus laevis, and skin and muscle cells, right? They can move around autonomously, like you're mentioning. They can be custom designed. This is really cool. Custom designed through computer algorithms. You can call them simple biological machines, and they've definitely caught the public's eye. And when it comes to the practical applications of these Xenobots, like what do you envision? This is something that's caught the public's attention. These self-assembling, self-moving, quote unquote biological robots. But for the practical applications of these things, what do you envision, and how do you think you can further improve on them?
Yeah, and by the way, I should say that the other really, we have lots of model systems in the lab, from ants and slime molds to human tissues, and in particular, planaria, these flatworms. We should at some point chat about that. But the Xenobots, okay, so there are, I see three basic lines of impact from these kinds of platforms, and not just specifically Xenobots. We're going to be making, and of course other groups are as well, making these sorts of things out of lots of different species and in lots of different configurations. First and most obvious kind of near-term applications are going to be specific, useful living machines. So once we really get better at controlling their behavior and their structure, you can imagine applications in the environment. So let's say biosensing, cleaning up plastics out of waterways, sensing or collecting rare biological molecules or other types of molecules. You can imagine using them in industry, anywhere that there's some sort of aqueous process going on. You can imagine using them in vitro for bioengineering. So let's say you wanted to microsculpt some sort of tissue for transplantation, right? That was too small. Make changes that are too small for a human microsurgeon to do. You could even imagine down the line, you could imagine functions in the body. You could imagine cleaning out arthritic knee joints, scraping plaque off of the artery walls, chasing down bacteria or cancer cells. Lots of potential applications. But I think that's kind of the clear and direct application. But I think maybe in the long term, maybe more impactful is going to be the use of this technology as a sandbox in which to crack the morphogenetic code, to really understand the mapping of what kind of stimuli and what kind of environment and what kind of signals do I need to give cells to build whatever I want. Because in figuring out how we program Xenobots for specific functions and specific shapes, we are going to really be able to crack that mapping and apply it to regenerative medicine in the patient. In other words, once we know, I mean, look, almost every problem of biomedicine except for infectious disease. So we're talking about birth defects. We're talking about traumatic injury, aging, degenerative disease, even cancer. All of these things boil down to one fundamental thing. How do you convince a group of cells to build whatever you want them to build, right? If we understood that, how to communicate to that collective intelligence to, I need you to build this kind of organ in this shape, in this location. Once we crack that, an incredible amount of biological issues are going to get solved. And so I see these biorobotic platforms as a way to really hone our understanding of that. Once we can make the Xenobots have whatever shape we want, this will be directly then applicable to tissues in regenerative context that have nothing to do with bots per se, but just to use that information to direct growth and form in vivo or in vitro for transplantation. And then I guess the third layer is really a little bit more philosophical, and it's simply the fact that we don't really have a science yet of predicting collective intelligence, right? When you have a group of subunits, would be the ants or humans or cells or robotics or Internet of Things or whatever, when you have a collection of these competent smaller units and we put them together into some sort of group structure, we really don't have very good ways of asking what kind of cognition is going to arise? Like, how sophisticated is going to be this group intelligence? What is it going to want? What is it going to be capable of? What kind of goals is it going to be capable of pursuing? We really don't understand this very, very much at all. And it's critical not only for regenerative medicine, but for really kind of existential kinds of questions for society going forward. So I see these platforms as starting to develop in a safe, very sort of circumscribed context, to start to develop that science of predicting collective intelligence that has the ability to solve problems.
I know we're all real precious individuals, but you're something else, Dr. Levin. You're a whole different kind of scientist, in my view. I mean, spent your whole career outside the box. I mean, born outside the box, with applied magnetic field on sea urchins. That's where it started, and you haven't looked back. And I mean, I think it's really about your exceptional creativity and the creativity of your lab members. And that is reflected in the website, which gives a really deep, I'm shifting gears a little bit here, but I was really impressed by your website. It gives a really deep insight into your research, the goals, your process, but also like delves into your scientific inspiration and gives a really, I think, nice and clear and comprehensive view of your thinking, of the way you want to approach this and the horizon you're reaching toward. It really clearly, to me, it took a lot of reflection and effort, or maybe not. Maybe you just break that off on a Tuesday morning, but to me, it looked like you spent a lot of time on it. Are you just trying to answer every possible question a potential postdoc might have while also inspiring them to join the lab? Or is this kind of just how you do everything? You think it all the way through first and then, get a nice coherent plan down?
Well, it's a combination of things. I mean, and I should say the website is quite a bit behind. I've got all sorts of updates in terms of conceptual things that have been developed conceptually in my group over the last year or so that need to be put up there. In particular, there's not enough focus on the cognition aspects of it as it is now. But one of the reasons I do this is because I want to communicate to people really the full impact of this road that we're on. Because oftentimes what people will say is, how come you do so many different things, right? I see artificial intelligence, and you get papers on cancer and behavior, and you've got the papers with all sorts of other sort of disparate disciplines. And why don't you focus? How come you do so many different things? And it's important for me to point out that these are not. I don't see these as completely different things. I mean, this is my version of focus, because I think we are actually all focused on one fundamental problem. All of these things are different ways to address a single problem, which is the scaling of mind in the physical world. That's what I'm interested in. I'm interested in how cognition is played out in various manifestations and embodiments, how it scales from little tiny proto-cognitive units that are basically just homeostats, and all they know how to do is keep to one sort of simple goal as a scalar parameter range, and how these things scale up into creatures that have second order high level metacognition and make claims about inner perspective that they have, and how they're not machines and this kind of thing, right? There's a lot of irony there in that journey. And so asking that kind of question in full requires lots of different disciplines. So computer science, physics, biology, of course, but cognitive and behavioral sciences. And all of our projects are somewhere in that. I mean, I make a lot of mind maps for our people internally, and sometimes for the website, showing how everything we do feeds into this larger effort to really understand how mind plays out in the physical universe. That's what ties it all together. And so, yeah, I like to put up. I like to give credit where credit is due. I mean, there have been incredibly brilliant people that have contributed to this idea, and some of them are on the website. Others we have, as you walk into our new Allen Center space, they're up on the wall in the main hallway. There are photographs of specific people with their names underneath, so people, when they walk in, they can see, what this is all based on. Just some, the brilliant folks who have gone before. So I like it because it inspires me, and hopefully other people find it interesting.
Yeah, definitely. Creativity is a group effort, right? And your lab is the perfect example of that. And it's been really so inspiring to catch up with you here, Dr. Levin. It's a very unique approach to developmental biology. I think the way what you're envisioning here and what you're doing in your lab, and I don't think we always get that perspective here on the show. I mean, we talk about a lot of cool new technologies and how ways to push therapeutics forward, but I think if we take a step back and really distill down what's going on at the cellular level and these intrinsic biological mechanisms, I think we can learn quite a bit more. So before we let you go, we're going to ask you a couple of peripheral questions, like, we like to do that here on the show at the tail end of our interviews. So first off, if you could answer any single scientific question, and you've answered quite a few of them, and you're attempting to answer quite a few of them, what would that question be? And it doesn't have to be in developmental biology. Regardless of your expertise or chosen field, what would that single scientific question that you would want to answer be?
Yeah, well, I consider myself extremely lucky and fortunate, because I work with people every day on the very question that I would like to most answer. And so I'm super happy that those two things aligned and I can actually do that. The question that I really would like to understand is how it is that mind scales up from physics. That's really what I want to know, and specifically where goals come from. I mean, I see cognition and intelligence as fundamentally goal-directed activities, regardless of whether they're made of silicon or evolved materials or whatever. But the origin of goals is a really profound question, and evolution is only part of the answer. And so that's something that keeps me up at night, kind of throughout since the time I was a kid. So that's what I'm really interested in.
Absolutely. And it's not just a basic science question, it's also in some ways a philosophical question too, right?
Yeah.
And so if there's I guess the last question we'll ask you is, if there's one hobby that you always wanted to pursue, but were never able to do it, what would you do? What would that hobby be that you would want to pursue?
The only thing I really always wished that I had done is, and hopefully at some point I will do, is study music. I find it amazing to think about, in terms of science and engineering. I understand the process. I understand how we get from where we are now to where we would like to be, and how you come up with ideas and how you implement them and so on. But the idea of being able to come up with a piece of music that others would find interesting to listen to is such in my head, as somebody who doesn't play anything, and my mother was a concert pianist, so you would think that I'd have a better grasp of it. But just that process and where that comes from and how it is that you do that seems just magical to me, and so that would be the one thing I would do, is I would like to learn to play some kind of instrument.
Well, Dr. Levin, you are certainly doing enough as it is without getting into the musical arts, but you're always welcome. And I know my co-host, maybe we like to talk about it on the show. He's an amateur guitarist himself, so maybe he could teach you a few things or get you on the bongos, whatever you need to do to fulfill that yen. We are on board, and we'd love to hear what you have to produce. Thank you, though, very much for sharing your scientific aspirations and knowledge and creativity, expertise, all of it. It's really been a treat and an inspiration. We really appreciate it.
Thank you so much. Yeah, really appreciate you having me on. Thank you for the discussion. Keep at it. It's great. Thank you.
All right, everyone, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or by email at info@stemcellpodcast.com with feedback or to suggest guests. When this episode airs, Arun and I will be in flight to the ISSCR 2022. Hope to see you all there. If you don't catch us, then you can always listen to us. We're going to be airing some special episodes. Hope you catch them. Until then, thanks for listening.